A warm greetings to one and all. I welcome you all to the session. In this session, we are going to discuss about the phase contrast microscope. So let me introduce myself as Dr.N.Aarthi, Assistant Professor, Department of Zoology, Sri Sarada College for Women. So in this session, we are going to discuss about the phase contrast microscope. What do you mean by phase contrast microscope and how it is going to work or the working principle of the phase contrast microscope? We know there are different types of microscope and how this phase contrast microscope is different from the other microscope. So two main features are going to play a major role in this phase contrast microscope. So let us move into the session and let us see in elaborate about the phase contrast microscope. So you can see the parts of the microscope first. So as I said, nothing new. It is there already what we have discussed about the different types of microscope. The two main parts which makes the difference is the face plate and the condenser annulus. These two are the important parts which is included in the light microscopy and which makes the huge difference and what we call as the phase contrast microscope. So what is this phase contrast microscope? So this is nothing but an optical microscopy technique that is going to phase shift the light which passes through the specimen to the brightness and changes the image. And it is mainly used in live specimens and living organisms, culture specimens, etc. and very thin specimens, thin sections likewise. So this is the minor or a small introduction about the phase contrast microscope, and it was first described in the year 1934 by Fritz Zerneck, and he was awarded the Nobel Prize for his invention by phase contrasting or by changing the diffraction of the light. It gives a huge difference in the image formation. For that, Fritz Zerneck was awarded Nobel Prize. So here two main important features are there which is going to play a major role in the functioning. So variation in the phase. One is variation in the phase. The light is getting variation in the phase plate. That is the first thing. And change in the amplitude which leads to the image contrast. Change in the amplitude or in the intensity of the light. Intensity is nothing but the amount of light entering or reaches the specimen. So based on that only the entire image is going to be created or formed. So this is the major thing what we have to highlight in this phase contrast microscope. So the different wave forms interacting with the different portions of the specimen. So here you have the specimen placed, and what it is going to do is the light or the source of illumination is going to reach through the condenser and then it reaches the specimen. Here what happens is different waveforms interacting with the different portions of the specimen. So you take an example, a cell is there and when the light reaches the nucleus, it is going to diffract. It is going to refract the indices. When the refraction and the diffraction takes place, the amount of light is getting contrasted. How it is going to take place? So that we will see in the principle, working principle of the phase contrast microscope. So these are the basic, this is the phase contrast microscope and these are the basic parts of the microscope, phase contrast microscope. That is the light source. As I said, the illumination will be there and the illumination reaches through the annular ring, then it reaches the condenser lens. From the condenser lens, it reaches the specimen, and here the light is getting scattered. Then it directs the light towards the faceplate. It reaches the faceplate, and then the objective and the image plane, and the image is formed. You can see the image. Okay, is it clear? That is phase shift. What to say, when the light reaches here, the direct light is going to create a phase shift. Based on that only, the image will be created. So how it is going to get created, we'll see here. So this is the working principle. So the lamp or the halogen lamp source is there, illumination is there, and it reaches the collector lens. Through the aperture, it reaches the condenser, and here you have the condenser annulus. As I said, two things are going to make a great difference in the microscope. That is the phase contrast microscope. One is condenser annulus and another one is the faceplate. These two are the major parts of the phase contrast microscope. And here from the condenser annulus, it reaches the condenser and then it reaches the specimen. So here from the specimen, you can see a different sort of image colors. You can see from here it reaches the halogen source gives yellow color light, and when it reaches the specimen, there are two different colors. One is orange. See, it is orange or red in color. Then it is having a yellow color. So this red source of light is called as diffracted light, and the yellow is called as the surrounded light or surround light. So what is this diffracted light and what is this surround light? Yes, the diffracted light is the light that interacts with the specimen. So you can see this is going to interact with the specimen and changes its phase due to variation in the refractive index. So due to the refractive index, when the light falls on the specimen, it is getting diffracted due to the high refractive indices. Okay. And what is this surround light? That is the yellow color light. The surround light is nothing but the light that passes through the specimen. So it is passing through the specimen without interacting much or diffraction. It is not getting diffracted. So might be those areas are thicker, might be thick or might be dark in area. So it is not getting the refraction. Okay. So what is the role of this faceplate? So what this faceplate is going to do? Yes, the faceplate is going to have or it is going to do bring together or combining together the diffracted light and the surround light are combined together with the help of this faceplate. And these combined light is going to interact together to bring the contrast. So they are going to bring a contrast and it is being imposed on the image plane. So both the red and the yellow together, they form a pink color face. So you can see here, that is the image plane. So now the contrasted light is going to form an image. Say for an example, as I said, when it is a cell, an animal cell, or might be a plant cell, whatever it may be. So when the light falls on the cell, the nucleus receives the light. So the nucleus will be dark. So there it is going to have a shift over. Since it is not stained, it is going to give a shift over, and here the image will be looking. The nucleus will be little bit dark, and the remaining portion will be little bit light. So you can give a contrast. One will be dark and another one will be light. So which needs more staining? Instead of staining, the light is going to give the contrast. So this is about the working principle of the light microscope. And what are all the advantages? What is the advantage in this finding? So without staining the specimen, you can see the image, and microorganisms can be seen, thin tissue slices can be seen, and subcellular particles including nuclei and other organelles can be visualized, and it plays a major role in the biological research. And what is the advantage? It makes a highly transparent object more visible, and no special preparation or fixation or staining is required. Okay, as such, we can go for the image screening. An examination of the intracellular components is relatively high resolution, which have the, say for an example, the dynamic motility of mitochondria, mitotic chromosomes, vacuoles can be examined in a very high resolution way. And phase contrast optical components can be added virtually to any bright field microscope, provided the specialized phase objectives. So pH, the objective piece need to be included with the phase plate alone. If it is included and the condenser annular is added, then it can be used to correct the size. And here also they have the limitations. So when compared to that of other microscope, it is often, how to say, based on the light and interaction and their diffraction only the image will be formed. So to use the phase contrast, the light path must be in an aligned way, and generally more light is required for the phase contrast than the bright field viewing or other things. So it has many other limitations also. So this is about the working principle or the structure of the phase contrast microscope. So thank you for listening to this video, and very shortly we will see or we will get connected with another video too. Thank you so much, and click on to the description link there. The link for the quiz or MCQ practice is given, and you can select that and get practiced about the phase contrast microscope. Thank you so much. Support by subscription. Thank you.